Did you know that the length of a DNA or deoxyribonucleic acid double helix in a typical mammalian cell is about 2.2 meters? This length is obtained by multiplying the total number of base pairs present in the DNA double helix with the distance between two consecutive base pairs. Now, the total number of base pairs present in a DNA double helix in a typical mammalian cell is 6.6 multiplied by 10 raised to the power 9, and the distance between two consecutive base pairs is 0.34 nanometers, or 0.34 multiplied by 10 raised to the power minus 9 meters. By multiplying these two factors, we get the length of the DNA double helix as 2.2 meters. Have you wondered how such a long polymer is packaged within a typical nucleus with a dimension of about 10 raised to the power minus 6 meters? To understand this, let us learn about the packaging of a DNA double helix, which is different for both prokaryotes and eukaryotes. In prokaryotes such as E. coli, the nucleus is not very well defined. However, the negatively charged DNA is not scattered throughout the cell, but is arranged in large loops, is held together by a few positively charged proteins in a region known as the nucleoid. On the other hand, in eukaryotes, the manner in which a DNA double helix is packaged within the cell is more complex. Eukaryotes contain a single molecule of negatively charged DNA packaged around a spool of positively charged basic proteins known as histones. The charge in a protein is decided by the abundance of amino acid residues with charged side chains. Now in this case, the proteins are positively charged as the histones are rich in basic amino acid residues like lysines and arginines, which carry positive charges in their side chains. Furthermore, the histones are arranged to form a unit of 8 molecules known as the histone octamer. Now around the positively charged octamer, a negatively charged DNA molecule is wrapped to form a structure known as the nucleosome, which is held in place by the H1 histone. A typical nucleosome has around 200 base pairs of DNA helix, and the nucleosomes present in chromatin can be seen as a beads-on-string structure when observed under an electron microscope. It is the nucleosomes that make up the repeating unit in chromatin, the thread-like bodies present in nucleus. Now, the chromatin fibers are of two types, euchromatin and heterochromatin. In a typical nucleus, euchromatin fibers of 30 to 80 nanometers in diameter are loosely packed and stain light. On the other hand, heterochromatin fibers of about 300 nanometers in diameter are more densely packed and stain dark. Moreover, of the two types of chromatin fibers, euchromatin is transcriptionally active, while heterochromatin is inactive. These chromatin fibers coil further and condense to form short and thick bodies called chromosomes during the metaphase stage of cell division. In this way, a DNA double helix is packaged into a chromosome, which is further packaged with